Hello, everyone. Good morning. Hi. Thank you all for joining us today. We're going to have some opening remarks, and then we're going to get into our first talk. Some housekeeping. This is a live and virtual meeting today, so we are streaming. If you have any questions for the presenters throughout the day, we're going to ask you to use the microphone, even though probably most people could hear you in this room. Or you can submit them via this Slido app. Every presentation will have its own little app, QR code, where you can ask questions to that presenter. Really excited to have you all here today. For those of you who don't know me, my name is Elise. I'm the VP of Research Programs at the Foundation. And I want to introduce our planning committee for today, Dr. Vishal Patel and Dr. Katarina Hopp. And we also had help from Dr. Ronak Lakia, who helped us go through the incredible amount of submissions we received for presentations and posters today. They'll talk more about that in a second. So let's go ahead and start with Dr. Patel.
All right. Good morning, everyone. Welcome, and thank you for joining us. For those of you who don't know me, my name is Vishal Patel. I'm a professor of medicine in the department at UT Southwestern Medical Center in Dallas. It has been an incredible honor to work along with Kat Hopp and Elise and Ronak to organize this meeting. The things that really excite me about this meeting are three things. First, most of the speakers are actually physically here, so you can actually interact with them. A lot of the talks, or in fact, most of the talks were chosen from abstract submissions. So these are original research presentations. Much of it is actually still unpublished. So we look forward to open dialogue, Q&A afterwards. And then third, as you'll see throughout the day, most of the speakers are young PIs or even trainees. So it's very nice to see the young crowd getting involved in the R&D day. So with that, let me pass over to Kat, who will tell us a little bit more about the agenda and how it's laid out.
Thanks, Vishal. So great seeing you guys here. And it has been a fun journey to organize this day, so I hope you will enjoy the outcome of how it looks after everything is said and done. The way this was put together by the PKD Foundation and us is that we kind of thought of what are the hot topics in PKD. We had a little bit of guidance just because we had FACIP last year, and so with the naming of the topics, there might be some overlap for the researchers that already see that, but they are secondly guided also by the focus areas that the PKD Foundation really wants to emphasize with their research programs. And so the areas that we ended up is focusing on the first session will be ARPKD and cystic diseases, thinking more beyond the actual most prominent form of PKD, which is ADPKD, as you all know, but we don't want to forget that other population that is also affected by this disease and where lots of great research is being done. And then the second session, after the poster session, which is from 11 to noon, is dietary intervention and metabolism. I think all of you appreciate that that is a rather hot topic, not only for the patient population, because they're very eager to use that information to be proactive with their lifestyles and changing the disease, but also from a research perspective, there's a lot done in how dietary intervention or metabolism at the cellular level drives the disease, even though it doesn't directly has impact on the PC1 or PC2 or other cystic disease genes per se. We then move on into a session that is called The Role of the Cystic Microenvironment. There was a similar session at FACIP last year. It is just one of these things that we wanted to ensure that we keep thinking beyond just the epithelium. The kidney is a very complex organ, so it is important to keep every other cell type or even extracellular vesicle that is floating within that interstitial space in mind as we do our research. And then we have the last 2 sessions being genetic medicine and cystic disease, which is really a rising topic to truly focus on how can we change the disease at the gene level, not the cellular level. And then in line with the PKD Foundation's aim, the translational and clinical session, to keep in mind that we're doing all of this in the end for our patient population. As Vishal mentioned, there's a lot of presenters that are truly also on the junior side of the things. We were really excited that we were able to invite to present today both of the outgoing PKD Foundation fellows to give their talk here. Both of them accepted, so we're excited about that. And they were invited speakers, and then the other invited speakers that typically speak at the first speaker of the session were just outstanding in their field and had a different perspective to their session. And so that's how they came into the program. Beyond that, we truly had over 70 abstract submissions, and as Vishal said, we had Ronak help us out to judge them and then select what we thought would fit not only best in the sessions, but were also the most outstanding abstracts to be presented in a form of an oral presentation. But all the others who were willing to come, please take your time during the poster session and go and look at their posters, interact with the presenters, and figure everything out along these lines. And with that, I give it back to Elise to introduce our keynote.
Let's give a round of applause to our planning committee. So I know we're all here to hear from a really fantastic, talented researcher. For 20 years, the PKD Foundation, in conjunction with the International Society of Nephrology, have given out the Lillian Kaplan International Prize for the Advancement of Polycystic Kidney Disease. Our award winner this year, Dr. Terry Watnick, is here to talk to us about the work that she has done over her career to lead to that prize. So let's not waste any more time, and Dr. Watnick, please come up.
Thank you, everyone, for coming. I'm only going to talk about part of the work I've done because it's kind of been all over the place, to be honest, from Drosophila to mouse studies to human studies. So this is one part of my story. My disclosures, none of them are relevant. I'll skip over the bio. And so first of all, I just want to really express my thanks to the Kaplan family for establishing this prize. To the Lillian Jean Kaplan Prize Advisory Committee for considering my application, to the nominators who are my colleagues who thought of me, and I appreciate it, the PKD Foundation, who not only supports this prize but has been really instrumental in my career over the past 20 years, and I'm really grateful for that. Probably my first grant was from the PKD Foundation, and then the International Society of Nephrology. So what I am going to talk about today, and the first is an introduction to PKD, but it's going to be brief, and I'm just going to highlight. I'm going to rush through it because I don't think I need to do it for this audience. I'm going to talk a little bit about the role of polycystins in vascular development, and then finally adult vascular phenotypes. So why did I get into PKD in the first place? Basically, I really thought at the point in my career when I started, which is I had just finished my clinical fellowship, clinical nephrology fellowship, I saw it as an opportunity to make a real difference. PKD, as I learned early on, is the most common genetic cause of kidney disease, affecting 1 in 500 to 1 in 1,000. It affects all races and ethnicities equally. And as you can see there in that picture, it's characterized by bilateral kidney cysts that cause progressive kidney enlargement. That is a kidney that was removed from a patient of mine that weighed 40 pounds, one kidney. And this, of course, leads to end-stage kidney disease, and it's the 4th most common cause of end-stage kidney disease. Usually, people arrive at end-stage kidney disease in the 5th or 6th decade of life, and certainly when I started, there was no FDA-approved treatment. So it really seemed like a place where as a clinician who might learn about science, I could make a difference. And that, of course, changed in 2018. And then the other thing about PKD that really struck me, and I started out in the clinic seeing PKD patients, was that although as a nephrologist I was focusing on the kidney disease, this is really a disease that has a lot of extrarenal manifestations. And I've kind of put those up here. There was a whole bunch of vascular phenotypes associated with PKD, and then cysts in organs other than the liver. Something that I've also worked on is the role of polycystins in male fertility, which is something that I think isn't that appreciated. So, you know, again, there's just like a lot of places where one can make a difference. And so this is going to be brief. I think this audience knows this, but the classic forms of polycystic kidney disease are essentially caused by mutations, by and large, in 2 genes. About 85% of people will have mutations in the PKD1 gene on chromosome 16, which encodes the polycystin-1 protein. About 15 to 20% will have mutations in PKD2, encoding polycystin-2. And then, as I guess we'll hear about and we all are aware of, there's a whole bunch of genes with the onset of genetic testing, we've come to appreciate that there are mutations in a whole bunch of other genes that can phenocopy PKD and can sometimes be confused for PKD. So this is where I'm going to summarize the work of a lot of people that has taken place over the past 20 or 30 years. And we have learned a lot about PKD proteins, or the polycystins, but I think, as you'll hear in subsequent sessions, there's really plenty more that we need to learn. So just very briefly, the product of the PKD1 gene is polycystin-1. It's a large membrane receptor, shown here, with 11 transmembrane domains. Multiple subcellular locations have been reported. Those are listed there. And also, speaking to the fact that we still have plenty to learn, there are many proposed functions for polycystin-1. A flow sensor, is it an atypical GPCR? It's been said to be involved in cell-cell or cell matrix interactions. As you'll hear in subsequent sections, also, is it a regulator of mitochondrial activity? Polycystin-2, the product of the PKD2 gene, has homology to TRP channel, to the proteins in the TRP channel family. Again, multiple subcellular locations reported, the endoplasmic reticulum, the cilium. Where is it really physiologically relevant? We still have work to do there. And again, in terms of its function, it's a non-selective cation channel. But recently we've come to appreciate, even though we thought initially it was just a calcium channel, this channel is also permeable to sodium and potassium. And most relevantly, it binds to polycystin-1 to form, in this very simple diagram, a receptor channel complex. And one thing which is going to come up later in my talk, and this is that cleavage is really important for the polycystin-1 function. And this GPS cleavage site was actually discovered by my colleague, Feng Chen, over there, and who has been a collaborator of mine over the past 20 or 30 years. So why are we even talking about the vascular complications of PKD? I mean, it all gets back to the patient, and I first got interested in this from seeing patients like the one I'm going to show you here. So this was a 53-year-old with no prior medical history that they knew of, and she presented to an emergency room with just like the worst headache of her life. In the screening lab, she had renal dysfunction, so they did a CT scan, and you can see here she has the classic findings of polycystic kidney disease, very enlarged kidneys filled with cysts. She had a head CT, had an intracerebral hemorrhage, went on to have an angiogram, which showed a large aneurysm right here of the left internal carotid artery. It was like 12 millimeters, and this thing probably had leaked a little bit. Anyway, she was very fortunate. They went on to be able to coil this, and she did okay. Didn't even develop kidney failure as a result of the contrast. So that's shown at the arrow there after they coiled it. So you see cases like this, and you start to wonder what these polycystin proteins must have a function in the vasculature if you keep seeing these kinds of manifestations over and over again. And so, you know, mouse models are what we typically use to try and answer these questions. I mean, it hasn't been that easy with mouse models. But what I'm going to show you here is some of the data that supports the idea that polycystins do play a role in the vasculature. So this is an older study by Dick Sanford where he introduced a LAC-Z reporter into the polycystin 1 gene. Okay, so you can do beta-gal staining, and everywhere that you see blue is a place where the polycystin 1 protein is expressed. And so this is very permissive for a role in the vasculature because you can see that PKD1 or polycystin 1 is expressed in all of the major blood vessels, and this is in mice, in the body, the ascending aorta, all of the major branches of the aorta. Here you have the descending aorta and the iliac arteries is expressed in the circle of Willis, or the, thanks, or the, these are the blood vessels at the base of the brain where most aneurysms arise, and you can see there's very strong expression of polycystin 1. And then in the heart, in the endothelium that lines the heart, and then the major vessel, the pulmonary artery. So if you look at the cellular level, and these are Western blots, this is work that we did. If you look at all different types of endothelial cells, you will see that both polycystin 1 up here and polycystin 2 are expressed, not only in blood endothelial cells, but in lymphatic endothelial cells. And then likewise, if you look at vascular smooth muscle cells, which are the other major cell type that make up blood vessels, you can see that both polycystin 1 and polycystin 2 are expressed. So this is very permissive for having a role, right? If there was no expression in those tissues, it would be unlikely to play a role. So then if you make mice where you knock out or completely delete PKD1 or PKD2, these mice do not survive. Okay, so it's unlike humans. They don't survive mid-gestation, which for the mouse is about embryonic day 14.5. And for people who have looked at these embryos, this is a normal embryo or a wild-type embryo. So here, the embryo is inside the membranes. And if you get rid of it, you get rid of these membranes. You can see here's the mouse. It looks normal. However, when you knock out PKD1, what you have is too much amniotic fluid, which is also called polyhydramnios. If you get rid of that, you can see this little asterisk shows that this embryo is very edematous. Okay? And then it's the same findings for PKD2 as you'd expect. And then with some embryos, you see a lot of hemorrhage. So these embryos are not surviving, probably because they have some kind of a severe vascular phenotype that's leading to intrauterine demise. Okay, so again, speaking to the fact that these genes do play a role in the vasculature, which we want to understand. So based on these kinds of observations and then other people's observations, the idea was that somehow these phenotypes were due to the role of PKD1 and PKD2 in endothelial cells. So what we did, and this is old data, was we used conditional mouse models and then deleted both PKD1 and PKD2 only in endothelial cells, with the idea that this would recapitulate the null phenotype. And so here we did this using a Tie2-Cre recombinase, which is just active in that cell type. And indeed, we did see that almost all of the embryos had polyhydramnios, which was a feature of the null mice, and you can see that there. I'm showing it to you for PKD1, but it was the same with PKD2. And then, however, if you get rid of this amniotic membrane, or you can see that there is no edema. So there was a significant difference. Whereas you do see edema in the null, we didn't see it when we just deleted PKD1 in endothelial cells. And there were some animals who had hemorrhage. And then we also found that there wasn't 100% lethality, but it was more like 30 or 40%. Okay, so that could be for many reasons. It's possible that the Cre recombinase isn't uniformly active. It's hard to say. So then we also looked at the placenta, because the placenta, especially the fetal placenta, is a very vascular organ. So, you know, maybe these things weren't sufficient to cause lethality. And what you can see here is that there's a very severe defect of the fetal placental vasculature. Okay, so what I'm showing you here is isolectin B4 staining of the labyrinth layer of the placenta and laminin staining, which outlines the blood vessels. And here in a wild-type embryo, you can see that these vessels there's a lot of them, and they're very tightly packed, which is what you need to have transfer of nutrients between the embryo and the mother. But in the PKD2, these embryos where we've deleted, in this case PKD2 in the vasculature, you can see that there's a real reduction in the vessel density, and also, I'm not showing this to you, but in the branching pattern. These vessels just don't branch normally. So we think that this is probably why in mice, if you completely delete all polycystins, why you have intrauterine demise. This is really probably a defect in the vasculature of the placenta. So we were puzzled, however, that when we deleted these genes in endothelial cells, we didn't really get edema, and we were wondering why that was exactly. So we went back to the literature and looked for other mouse models that also had edema and that looked very much like PKD1. And when we looked at this, what we realized is that mutants in other mouse genes all had to do with the lymphatic vasculature, okay? So I'm just taking some data from the literature here. Here's a PROX1 mutant, and PROX1 is involved in specification of lymphatic endothelial cells. Here's VEGF-C mutant. You can see it has edema. That's involved in migration of lymphatic endothelial cells. And then again, TY1, these embryos have edema, and this gene is involved in lymphatic endothelial cell survival. So this gave us the idea, okay, like why don't we just go back and look at the lymphatic vasculature in these PKD null embryos, which is what we did. And one of the really good ways to look at the lymphatic vasculature is to look at it in the skin of the developing embryo. And that's what I'm showing you here in both. Here's a control embryo where we're looking at the skin at E14.5, which is the time when you get fetal demise, and you can stain with endomucin and LIF1. LIF1 is a marker of the lymphatic vasculature. Endomucin in red is just like all the other blood vessels, and you can see here that you have a very nice network of lymphatic vessels. They're not dilated, they look like normal. However, in both the PKD1 and the PKD2 mutant embryos, these lymphatic vessels are there's not many of them, they're not branched properly, and they're just like grossly dilated. Okay, and over here, I'm actually just showing you the quantification of that. Here is the density of lymphatic vessels in the mutants. You can see it's decreased. And here is the diameter of the vessels. It's increased in the null, and then branch points are decreased. So there's some problem with the way these cells are branching, and they're just becoming dilated, and this is probably why these embryos are edematous. We also looked at a bunch of other lymphatic developmental markers, which I'm not showing you here, but every place we looked, it looked kind of aberrant. So one thing that we were asked is, well, okay, how do you know that this edema you're seeing isn't really the result of a cardiac defect as opposed to a vascular defect, because that's also possible. So, and in addition to that, conotruncal heart defects, which are developmental heart defects, had been described in some PKD1 and PKD2 mouse models. So what we decided to do is to do micro-CT of these embryos to visualize the cardiac structures. We did this only for PKD1 because it was very costly at the time, and we looked at E14.5. All these embryos had edema, but we were unable to find any discernible cardiac defects in these embryos. So that's not to say that they don't occur, they may, but we concluded that edema does not depend on the presence of cardiac abnormalities and really speaks to a primary lymphatic defect. Okay, so then because we had seen these branching defects, which I'm not showing you all of this, we in order for vessels to branch, you have to have normal cell migration because, the cells migrate forward to branch. So we decided to look at this in vitro, and unfortunately, if you go to isolate endothelial cells or lymphatic endothelial cells from these knockout embryos, you have to use a bead technique, and the beads stick to the cells, which kind of completely disturbs migration. So we had to find a different way to do it, and what we decided to do was to use shRNA to knock out both PKD1 and PKD2 in lymphatic endothelial cells, the human lung cells that we just bought from a company, and these are primary cells. And so here in the top panel, we used 2 different shRNAs to PKD1. We were able here on the Western blot, you can see it's not complete, but we have pretty good cell knockdown. And then you scratch the plate, you let the cells migrate, and you can see that the wound does not heal as efficiently in the knockout as it does in the wild type. And the same was true for PKD2. Here's our PKD2 Western blot, pretty effective. So there was a defect in cell migration, and this had been reported by Alessandro Valletta's group for other cells as well. And so then we decided to look at whether directed cell migration was defective. Okay, so in order to characterize the migratory phenotype of these polycystin-depleted lymphatic endothelial cells in more detail, we tracked individual cells in wound healing assays with time-lapse video microscopy. And so what you can see here, each of these lines is one cell. Okay, so and that's the track. So if this is the wound, you can see that in the wild type, they're all kind of going directly to fill the wound. And in PKD1 and PKD2 deficient cells, it's just like much more random. So if you look at the total accumulated distance that the cells are traveling, it's not any different between wild type and knockout cells. But if you look at the Euclidean distance, which is like going forward, you can see that a lot of these cells are just going in the backward direction. So there's a defect in directional cell migration, and we did some other work, which I don't have time to show you here, that kind of confirmed that finding. Okay, so we were wondering at this time, it seemed, okay, so like we have this lymphatic cell defect, I don't know if it's true, is it just us that are finding this? And I just want to show you this data because it just happened serendipitously that at the same time I was contacted by an investigator in Australia who was a lymphatic expert, which I was not. And this investigator was doing a zebrafish screen for genes that cause lymphatic defects, and just doing this screen without, any bias, he actually identified a truncating mutation in one of the zebrafish PKD1 genes, and there are 2. And it was in this PKD1A, and you can see this here. So this is a wild type fish, and right down here is like where you would normally see the lymphatic vessels forming. But in this mutant, which he called LIC1, and here that's the mutation, it's a truncating mutation, you can see that the lymphatic vessels are not like branching. They actually branch off of some of these veins, and it just doesn't happen properly. So it does seem, putting it all together, that our polycystin genes do play a kind of a critical role in vascular development, and particularly in lymphatic development, and this seems to be conserved across other species as well. So now I'm going to go to a little bit of unpublished data, and I'm going to credit all of this work to Feng Chen, who's sitting there. As I told you in the beginning, polycystin-1 undergoes an autoproteolytic cleavage event, which was characterized by Feng, happens in the ER at a GPS cleavage site, which is indicated over there. And these 2 halves of the protein, which Feng has named the NTF and CTF, remain non-covalently associated. So Feng designed a mouse where he mutated that cleavage site, and he replaced a T here for a V, and he generated this mouse line, which he has called the VV mice. And mice lacking this cleavage site actually survived to about 3 weeks of birth. Okay, so they're born, and they don't have any apparent embryonic abnormalities, but as shown here, and this is from Feng's paper, they do go on to develop renal cysts in the postnatal period, and eventually they do die. So we were kind of really interested in this and the fact that these mice survive, yet there's a pretty severe kidney phenotype, and we decided to go back in collaboration with Feng and look at the embryonic phenotype of these mice and see what was going on. And so this is showing some of this data. Here's a wild type mice, and here's the null phenotype, which I showed you before. With polyhydramnios, we've quantified the amniotic fluid, and you can see in the null it's just a lot higher, and edema. And when we looked at these VV mice, as you'd expect because they're surviving, they had no polyhydramnios and no edema. And the quantification for the amniotic fluid is shown here. It's like not any different than wild type. We went a step further, and then this is in collaboration with Fung and the Yale O'Brien Center. They made a BAC transgenic that had this VV mutation introduced into the BAC, and we went ahead and crossed it into the null phenotype and into the null mice, and sure enough, this completely rescues the vascular defect, and that's shown here. So you can see these mice don't have any polyhydramnios and no edema, and that is just a Western blot showing that, yes, in fact, this VV BAC is made, and there's no cleavage going on here. So that's just confirming at the Western blot level that this is the case. So yeah, so then we went and looked at the skin, and as you'd predict based on what I showed you, you would sort of expect that VV would have no lymphatic defects. And again, that is shown here. You can see this very nice, this is again LIF1 staining, endomucin staining. You can see that the lymphatic network is just all nice and looks no different than normal. And we can also, again, in the null background, by introducing this VV back, completely rescue that lymphatic defect. And that's shown here. And here is the quantification of all of that. Okay, so the one thing I didn't tell you is that this cleavage event is very necessary for the polycystin complex to traffic to the primary cilium, right? So, and that's thought to be really critical for averting any type of cystic kidney disease. So we thought, well, okay, like what's going on in the endothelial cells? Are you seeing polycystins traffic to the cilium or not? And here's our data. For staining, we isolated endothelial cells from E14.5 embryos. And here is a wild type back. So we're using HA staining because, as people who work in the field know, the antibodies just don't work that well for immunohistochemistry. So we had to find a different way of doing this. So we're using backs that are tagged with HA. And what you can see here is that in the wild type, the polycystin 1 is going to the cilium, as you'd expect, and that there's RL13B, which marks the cilium. The same is true for polycystin 2. I'm just not showing it. And here, with the VV back, in the null background, or you don't even really need to use a null background, but polycystin 1, it's not cleaved, none of it traffics to the cilium. So what this is telling us is that this full-length polycystin 1 that's not cleaved must have some important role in some cell types outside of the cilium, which I don't think has been reported before. And we're really interested in this and are continuing to pursue it to try to figure out where in the cell that might be. So I've now told you a little bit about embryonic phenotypes, but I'm not sure what the relationship between all of that is and the adult phenotypes that we see in our patients, right? Like, how does that developmental branching relate to the formation of an aneurysm? I think it's not so clear. So that's been another effort to try to think about that and figure out how to address that, which is really hard. Actually, it turns out because these mouse models don't really develop a vascular phenotype. So we're kind of trying. But I guess that tells us we got to go back to our main model organism, which is people. And so, we have spent, oops. Okay. I didn't do anything. What should I do? I don't think I touched anything. The computer's just going on and off.
Just try restarting.
Okay. Okay, I'm going to try not to touch anything. I just want to go back to because I always think you want to, as a physician or MD, I'm always going back to what do we learn from human patients? And so this is a summary of some of the observational studies. There have been more now. And I think that this gives us, when we look at these studies, gives us some important clues about some of the things we need to think about. And so what I've done here is summarize studies where they were screening all comers with PKD for brain aneurysms. Okay, and so there you see, I'm afraid to touch anything, but okay. Here are the numbers screened, and this is the percent of, if I took 100 people with PKD, just screen them, what percent would have a brain aneurysm? And what you can see here is it's somewhere around 10%, okay, give or take. And these are retrospective studies in some cases, so it's kind of hard, but I think that most people would agree it's about 10%. In the general population, if I took 100 people out on the street, it would be somewhere around 2%. So there's definitely an increased risk, okay? It's about maybe 4 to 5 times the risk of the general population. However, if you then divide people into those that have a family history of PKD and aneurysms and those that don't, you can really see a difference. And so if I take those that have a family history, and of course that's assuming that people always know their family history, it's somewhere around 20 to 22%. So you can see that a family history of having a brain aneurysm in PKD is like a huge risk factor, and that risk is further increased by about 4 to 5-fold. If you then look at those people who don't have a family history, it's more like about 5 to 6%. So you can see that there's a real difference. And what this really tells us, or has told us, is that there's some genetic component to this that I think if we could understand would be great, would help patients, might inform how we screen, et cetera, et cetera. So thinking about this, like, okay, like what would make someone have an aneurysm, I've put there the 3 things that you can think about. One is just environmental factors. So even in no matter PKD or not, smoking is a risk factor for an aneurysm, uncontrolled blood pressure, and excess alcohol use are the ones that have been identified. Those things are kind of difficult to capture, but the 2 things I think we maybe can get a foothold in are the primary mutation. Is there something about some PKD1 or PKD2 mutations that predispose to this? And then I think the other major category, and I'll address them separately, are genetic modifiers, which I think most people think can impact kidney disease as well as probably other phenotypes. And so I'm going to just start out with the germline mutation issue. We are not by any means the only people that have looked at this. I mean, Peter Harris has, and as well as Emily Cornick-Legall, but I'm just going to focus on the data that I did, because this is something I did as a fellow very early on because I'm just interested. And we did, with very rudimentary techniques at the time, look at the germline mutations in a population of patients that had aneurysms or severe onset disease. And this is a collaboration with the group at Colorado at the time. Patty Gabow sent us all of her samples. We looked at them. And we were just surprised that we found 3 families that were unrelated with the exact same mutation. So, you know, at the time we thought, well, maybe this was by chance. We weren't really doing full genome sequencing, but always this stuck in my mind as why this might be. And this is a common mutation, so there are all kinds of reasons why this might have happened by chance. But at the time, I was always interested in this aneurysm question. I had gone back to the literature to look to see what kinds of families we could find. And this is a report of a family that was done at the time by Steve Somlo, believe it or not, when he was like probably a fellow, I think, or not much past it. And this family was really interesting because they had a co-segregation of a connective tissue disorder with aortic aneurysm dissection that looked kind of like Marfan syndrome, and also polycystic kidney disease. And those blue circles I'm showing you here are individuals who had PKD, classic PKD, but they also had thoracic aortic aneurysms with dissection. And then there were some other people here who maybe didn't have aortic dissections but had other features of overlap connective tissue disorder like arachnodactyly, which is long fingers, joint laxity, pectus deformity, et cetera. And so we managed to get the DNA from this family. Somehow we wrote to the doctor who is the last author on this paper. He sent us the DNA. We did gene testing, and sure enough, this family had that exact same other mutation that I talked about. It was just by chance. And again, it could all just be chance, but I have always wondered about it. And so one idea I had is that maybe this isn't just this mutation, but it's a type of mutation where this mutation actually results in a frame shift. It's a 2 base pair deletion, so the protein is frame shifted, but it doesn't end right away. It actually adds on a number of amino acids, like 98 amino acids afterwards. So theoretically, this could either just never get made. I mean, you could just go nonsense-mediated decay, no protein made. But if it were to be made, it could be secreted, and then it would presumably be secreted into the matrix. And one thing that has been known about aneurysms is that there's some disturbance of the matrix, right? This is weakened matrix, and that's why you get aneurysm formation. So I always kind of wondered about this, and Peter Harris has always said, oh, it's just because it's a common mutation, there's nothing there, you know. But anyway, I didn't give up, and I just, and this is a collaboration with Asha Magulati and Steve Somlo, we decided to make a back transgenic mouse that had this mutation engineered into the human copy of PKD1, because the mouse gene isn't so similar at the DNA level that you would be able to replicate this. So that's what we did. We made this back transgenic, we crossed it into the PKD1 mutant mouse, and it does not rescue at all. Okay, so it's definitely a mutant protein, and you can see that here at E14.5, these embryos don't survive. I'm not showing you that data, but they don't. They have polyhydramnios edema. So then we wanted to know, well, okay, so it doesn't rescue, but is it still made? It's possible. And sure enough, we looked at Western blot, and this is the Western blot showing you that here, that this gene makes a protein. It's not functional, but it is made. And then we looked to see whether, well, it could just be degraded, made into protein, and not really ever get trafficked. But we did, and we have a lot more data, which I'm not showing you, but we do think that it might traffic. So when we look, this is just from the total embryo. Here is the untreated protein. If you treat with PNGase, you take away all of the glycosylation sites. But there is some endo-H resistance, suggesting that it does traffic past the ER. And we're still trying to figure out what this does. Does it get out into the matrix, for example? I don't know. We'll see. And so now I'm going to move on to the last, the one determinant that I still think we need to address, and it's a hard one, is whether there are any modifiers that could interact with the PKD1 and PKD2 mutation and lead to this phenotype, which I think is quite likely, actually. And so the way we kind of got into this is, again, in mouse models, trying to figure out if this is even something that's possible. And we then, because of that family, got stuck on this issue of Marfan's and whether there could be some interaction between fibrillin-1, which is the gene that causes Marfan's. And just to remind you, because you probably may not know this, but Marfan syndrome is an autosomal dominant connective tissue disorder, okay? It's caused by mutations in fibrillin-1, which is a matrix protein. And aortic aneurysms in this disease involve the ascending aorta. You can get mitral valve prolapse. You can get brain aneurysms, though less commonly. And there's a whole bunch of these other phenotypes that are associated, including arachnodactyly, as I told you, in that PKD family, long limbs, pectus, where you get like a curvature of the sternum, joint laxity. And this has been shown now by Hal Dietz's group at Hopkins to be, or he thinks it's caused by increased TGF-beta signaling, okay? And that's just shown here. This is taken from one of his papers looking at phosphorylated SMAD2 signaling in the ascending aorta, and you can just see that it's going to be nuclear when it's active, and you can see that in their fibrillin mutant mice, that you just have a lot more TGF-beta signaling. So I don't know, we were at Hopkins at the time, so we went to Hal, and we just said, okay, let's do the Hail Mary experiment. We'll just cross our mice and see what happens. And that's, in fact, what we did. We took heterozygous mice of each type, crossed them together, and then we were going to look at the mouse that had mutations in both genes, okay? So we didn't expect anything, but we were kind of surprised that there was indeed some interaction between these two genes. So here I'm showing you VVG staining, which outlines the elastic lamina of the ascending aorta. Here's a wild-type mouse. Here's a PKD1 heterozygous mouse. And these mice are like 6 months old, so it's not a rapid thing. And what you can see here is that the elastic lamina are just like very nicely organized. The aortic wall is thin, just fine. And then in the fibrillin heterozygote, it's not quite that way. The fibers are not, they're kind of broken up. The aortic wall's a little thicker. But in the double heterozygote, it's just a lot worse. And this is kind of a gradual thing. It was a hard study to do because it really took like about 6 months. And Hal developed this aortic architecture score, which we had graded. Aram, if she's here, I know helped with this, doing this blinded, just looking at the aortic architecture score, and it's certainly worse in the double heterozygote. We then looked at TGF-beta signaling with the idea that somehow PKD1 was making this worse. This is ascending aorta at 6 months, and you can see that in the double heterozygote, there's more nuclear phosphorylated SMAD2, suggesting that there's a further upregulation of TGF-beta signaling above what you would see in the fibrillin-1 heterozygote. We also looked at CTGF, which is a TGF-beta downstream gene, and it looked like there was more of it activated in the double heterozygote. And this is the quantification. Then we said, okay, well, like we did it that way. What if we completely deleted PKD1 in vascular smooth muscle cells? Would we see anything? And it was not quite as dramatic. And so here you have the wild type, and here is where we've deleted PKD1 in the vascular smooth muscle cells. And this is just looking at ascending aorta. And what you can see here is that you have disruption of the elastic lamina. The aortic architecture score is worse. And this took 6 months. It's not fast. It's like you have to wait 6 months to see this. We then also did aortic measurements by echo and ultrasound. And in fact, we saw a dilation of the aortic root in the ascending aorta in these mice, which these mice didn't die. So I don't know clinical significance in a mouse, but it was statistically significant. We then looked at TGF-beta signaling in the ascending aorta. And here, what we're looking at is we immunoprecipitate with SMAD2/3 and probe with SMAD4, because when TGF-beta is activated, SMAD4 is recruited to SMAD2/3, and you can see that there's more of it in the ascending aorta where you've completely knocked out PKD1. We looked at CTGF, and again, you can see that there's more of it in the PKD1 vascular smooth muscle cell deleted mice. And that was all quantified. So, we're dealing with ascending aorta here. You could say, like, well, what does that have to do with aneurysms? I do think they're linked, but so coming back again to a patient, this is someone I saw. So, like people say, like, well, can you get aortic tissue from patients and look at this? And it's just obviously hard. But this was an 18-year-old. She had a family history of very classic PKD. And when she was 18, she was a college student. She kind of showed up with very bad hypertension. And the student health services, rather than sending her to a nephrologist, actually sent her to a cardiologist. So, of course, you have a hammer, everything's a nail. The cardiologist did a cardiac echo. And rather than a renal imaging, which she eventually had. But she did have PKD, but she had an ascending aortic aneurysm. So her ascending aorta measured 4.7 centimeters, which for an 18-year-old, that's very abnormal. It's not normal at all. And so she was followed, and here's the MRI showing this dilated aorta. So she was followed for a while, but then this thing kind of continued to dilate, and they decided that they were going to preemptively repair it. And so she had this repair done when she was 19. And so we were fortunate. We asked the surgeon, can we have like just a little bit of this aortic tissue, which we did. And this is staining to look at the matrix. And what you can see here is this is a control aorta from somebody else, and you can see here that there's some expansion of the matrix. It's not normal. We then, of course, and this is in collaboration with Hal Dietz, wanted to look at TGF-beta signaling. We didn't have enough to do a Western blot, but we stained with SMAD2, phosphorylated. This is Loeys-Dietz, where you have really massive upregulation of TGF-beta just as a positive control. And you can see that it's increased over the control, but not quite as bad as Loeys-Dietz. And this, of course, is an N of 1. There's no way to do any kind of statistics on this at all. But and so I don't know that this is what happens in people, but I think the message is that it's very plausible that genetic modifiers could intensify the risk of aneurysm and PKD, and there's several types of things you could envision. There could be rare coding variants in structural genes for vascular integrity and other vascular signaling pathways, or maybe TGF-beta pathway components. Or there could be common variants, and there have been a number of loci identified in GWAS studies of sporadic aneurysm, so could there be an additive effect? And so I just want to put in a plug for this. We're trying to collect a very well-characterized cohort to look at this issue. It's a remote study. We don't need you to come anywhere. We can do it by phone and by mail. So far, we've collected close to 300 individuals with brain aneurysms and PKD, but we need more. So if you have any cases you can contribute, just contact me. And so in summary, autosomal dominant PKD in humans is associated with a vascular phenotype. I think that's clear, with intracranial aneurysms being the most common and lethal manifestation. Mouse models have shown a developmental role for polycystins in the vasculature, including a novel function in lymphatic development. Contributions of genetic modifiers, environmental factors, and potentially some germline mutations to this vascular phenotype in ADPKD is possible. And I do think that a better understanding of these factors could lead to more robust screening algorithms. And then finally, I still think we need to remember, and this goes for all of the extrarenal manifestations of PKD, I think it's impossible to make important discoveries about polycystin biology by looking outside the kidney, and we shouldn't forget that. And so I just want to, just a few acknowledgments. I need to say that most of the work I showed you was done by Dennis Baskin and Patricia Utera, who many of you know. And then I have just had great collaboration over the years with Feng. We've worked together since we were playing in the sandbox. My collaborators at Yale, Steve Somlow, Whitney Bessie, Ashima, who's sitting here, has done a lot, Neera Dahl when she was at Yale, my Hopkins collaborators, and then all of the various people who have contributed samples. If I missed anyone, I'm sorry, but we have a lot. We've gotten help from a lot of people in this, and I'm appreciative. And then I just want to say that I've been so fortunate in Baltimore to work with people. A lot of you are sitting in this room and can probably see yourselves in these pictures here. We started out at Hopkins with Greg Germino founding our group, and then we have subsequently over the years grown and contracted. We're now at the University of Maryland, and here's our University of Maryland Cystbusters team at our PKD walk. And I just feel very fortunate to have had the opportunity to work with so many great people. And so I'll end there.
Thank you so much, Terry, for this fantastic presentation. We have time for maybe one, maybe two questions. Yep.
Some years ago at a conference, I asked you the question about the potential role of overexpression of MMP1 and MMP9 with respect to not only PKD, but the extracellular manifestations, cerebral aneurysms, aortic aneurysms. I'm just wondering if there's been any development in terms of your thinking in that regard.
I mean, it's certainly possible, but I haven't formally looked at it.
Hi, Terry. Latent TGF-beta is converted to active by alpha-V integrins. We've seen abnormal distributions of alpha-Vs in kidneys, cystic kidneys. Have you looked in the muscle vascularity for that?
I'd be really interested in that, because that is exactly what is the mechanism that's postulated in Marfan's, right? Because fibrillin is an extracellular protein, and when you get mutant protein, you have increased activation, there's more availability. So yeah, that's certainly possible.
Terry, congratulations and great talk as well. I wanted to ask if you had expressed your truncated back on an adult conditional model to see if you can induce a
Yeah, we're doing that now.
Great.
Yeah. Yeah, there's a lot of breeding, yes.
There.
Yes, we've done it. We have to wait like 6 months, I think, and we're doing it.
I said 2, but last question, number 4.
Thanks so much. Great talk. As kidney is a very vascular organ, do you think that this polycystin expression in the vessels contributing to cyst formation or PKD progression in the kidneys?
Yeah, it's a really good question, but I think the issue is in the human situation. Presumably you're heterozygous, right? So you probably would have to have full loss in a blood vessel to really, I don't know that heterozygous loss is sufficient. That's the question.
And the lymphoid cell migration, do you think is it associated maybe with the immune functions of this patient?
Yeah, I think that, and even in aneurysms, immune activation could be a part of it, yes. But I don't know. I'd like to collaborate with someone if anyone wants to look.
Let's thank Terry again for a fantastic presentation. It's time for a break now, and we reconvene at 10.